Okay, so I spent some time today and yesterday grading your homework from 1 to 4. Most of you did very good job, and I'm happy to see how the class is progressing and how you're learning. Some of the homework, maybe 1 or 2, I could not upload it because as you were told, you submitted maybe some PDF files and some other files, which I believe ePoly may have some limitation. So when the file sizes are getting large, I'm not able to just at least upload it. So there are only few instances. So if you received a comment or I cannot see your homework, maybe you can make another attempt, and there you can only submit the text file, the script as a text file. Other things I have seen people do, they usually zip it. So once you zip it, I'm able to upload it, and it may contain several files. But at any rate, if you received a comment saying I cannot grade your homework, just resubmit the script only. I don't need anything else in your Blackboard, so I can grade it. So that leaves us with homework 5, 6, and 7. So homework 5 on hypergeometric is due next week. I'm posting the gene set enrichment analysis homework after today's review tomorrow, which will be due 2 weeks from now, around end of November. And your final homework, Limma, linear modeling, which I believe personally will be the most difficult homework that you have done to this date. It's a hard concept for me also. So that will be due around December 10th or so. And then you will have your project that will start proposing and reviewing with us. So the last 2 weeks of December, I would have you guys present to the rest of the group. I received a proposal from group J, and group Y I think are going to resubmit. You guys had some question. I haven't had any project proposal for group O. So remember, project is the most important thing in your grade, which can override the grade that you may have had on your homework. So if you haven't submitted your project proposal by tomorrow, which is the deadline, November 15th, please do so. So for meta-analysis, as long as you're working with 3 data set, that's more than enough. So just so I know that you know the concept. So those were some of the logistics of homeworks, how we have graded so far, where you stand, and what are the remaining 3 homeworks that you expect, and what are the remaining topics for the last few weeks that we have in the class. Okay. So let me get started with gene set enrichment analysis, and I believe there is no question up till this point. Okay, so to understand gene set enrichment analysis, you will need to read chapter 13 of your book. There is a dedicated chapter on gene set enrichment analysis, or GSEA. So I have taken some notes, and I will try to explain to you to the best ability that I can. And also on lecture 9, I give you the guidance, so you should really take a moment and upload this paper at gseamuta.pdf and read. That's where the work originally started, and I know personally that this is extensively used, this methodology in microarray. So it's very useful. It's one of the techniques that you should know and should be comfortable working with. So read the paper, and you'll probably understand it better than I'll be able to explain you today. And so that's your first starting point. So today I will go over the GSEA package like I usually do with the ALL data, and I have some notes here along the way to help you understand. So this stands for gene set enrichment analysis. So what's the basic idea behind it is you use a predefined collection of genes, and that you try to relate to your experiment. So what we have done up until this point is we start with, let's say, 12,000 features in an array. Then we do some non-specific filtering. Then we do t-test, SAM, multi-test, whatever you want. And then you're selecting the top 10 or top 15 genes, and you're trying to map it to a pathway. So that's so far what you have done so far. Now you got exposed to a different idea on the hypergeometric test, and I give you an example of in an urn you have balls, red and white and black, whatever. So now that you did your analysis and you pick like top 10 genes, so you need to do an additional testing to say what is the probability of me picking those 10 genes or 10 go terms just by per chance, or there is some real significance to those go terms that you're picking from the urn. So you then do your last bit of testing by hypergeometric testing. You construct that and you try to establish a p-value, and then end of the day you try to conclude that those go terms or those 10-50 genes that I have picked that maps to a go term are really significant based on the p-value and hypergeometric test that I'm doing. So that's one way of thinking of it, and you're already working on homework 5 with hypergeometric test, and chapter 14 on your textbook. GSEA takes you from a different angle. Here you actually pick some predefined pathways or predefined go terms, and that you try to compare with your genes that you have found out to be top expressed in your experiment, and try to relate them. So maybe later on, as I show you a little more diagram, it will become clear to you. So the basic idea is, first of all, you define what are the pathways that you want to work with, and then you try to develop a technique to say, are these pathways that I'm pre-selecting have any relation to the experiment that I just did? Because most of the time a priori, you have a pretty good idea of the biology that you're doing for your experiment. So you pick your pathways, and then you try to say that those set of pathways that you have picked, which one has the most likely correlation based on how the genes have been expressed in your experiment. So that's the idea. So it's different than what you have done so far. You don't define a cutoff value and try to say these are my top 50 genes. So it's different, right? So because you start with your predefined genes. And what you do for each of those pathways that you have picked, you try to assign a score. And we'll go over the score, how you do that. And whichever pathway comes up with the best score, so then you say, that's the pathway that has the most significance to my experiment. So simply say it again. You can say that I'm going to pick 10 pathway, and I'm going to compare each of this pathway to my microarray experiment. And for each of this pathway, we'll use a technique to derive a score. And whichever pathway gives you the best score, then you can say, this is the pathway that I'm interested in, and my experiment tells me that this pathway has been impacted the most. Little different. So let's try to visualize this from the diagram that we had in the paper. So what you were doing, and let me maximize this. I don't know whether 200% will be better or not. Maybe. So what the paper tells you that you start with a collection of gene sets, right? So those collections could be a bunch of genes in a pathway, it could be many genes in a go term, or you can have some cluster of genes that you have based on past experiments you have done. When you did your hierarchical clustering or other clustering, you may have seen a lot of patterns where a bunch of genes are getting clustered together. So that can be also your choice, or you can just use pathways like it would be in the KEGG database, or your company may have proprietary data on pathways. So either way, either of these you can use, but all they are doing is a collection of genes under a pathway or under the go term. So you start with that. Then in the paper, they did an experiment with two types of samples. One was NGT, was normal glucose tolerance, and the other one was type 2 diabetes. So as they did that, they ranked the genes R1, R2, R3, R4, etc. from high to low expression values. And the other one is they picked, let's say, 5 or 10 or whatever number of pathways, and they go through each genes and they mark, is this gene R1 part of my first pathway P, right? You can have many pathways, so you evaluate each pathway at a time. So let's say P1, and then you go through the genes to say, is R1 a member of my P1? No. R2 a member of my pathway? Yes. So that's the first thing that you do. Then for each of this pathway, knowing what gene is part of that pathway and what gene is not part of that pathway, you derive an enrichment score. And those of you who want to understand the statistics behind it better, it's derived from Kolmogorov-Smirnov, the running score enrichment score statistics. So that the basic algorithm came from them. And what then again we are doing, now that we have each of those pathways and we have our genes, we know whether this gene is present or absent, then what we do is, let's say if the gene, and we will see a different implementation, but the idea is same. So the way I would read this running sum, so let's say R1 is not part of your pathway 1, so you get a negative score. R2 maybe is a part of your pathway, so you get an up score. And this value of how negative it is or how up it is, you can use the original t-test that you used. So as more and more genes are getting part of your pathway, so you're adding, so you're going up, then maybe more genes were also not part of the pathway, so you go down. So that's the running score. Like sometimes it will go up, sometimes it will go down, and how much it goes up or how much it goes down may depend on the p-value or t-statistics that has been calculated for each of these genes. So you will get a graph like that for each of your pathway, and the peak, the highest peak of your graph is the enrichment score. So for each of those pathways, so if you have the pathway 1, it may have a plot like this for your running score. So that gave you an enrichment score. So the highest peak is the maximum enrichment score. So for each of the pathway that you do, you want to record the MES for the actual perfect data. Then just because you have 10 pathways and 2 of the pathways told you that you got a maximum enrichment score of 346, it's not end of the story. Like we always do, you have to actually then permutate the class label. You have to get a reference, then you have to get a reference distribution based on how you may have got this maximum enrichment score just based on chance, right? So what you have to do, the actual maximum enrichment scores that you got for each pathway, you have to compare that with a permutated data set. And you have to establish a p-value cutoff. So if your permutated database enrichment score and the maximum enrichment score that you're getting on your actual data set, if those are actually standing out, that even if you did permutation, you're not going to just by chance get an MES of 346 based on your p-value cutoff, then you can just say, this pathway with a maximum enrichment score 346 doesn't seem like I got it by chance. So that's the final cutoff you will do on your statistical analysis. So then you will actually start focusing on your OXPOH pathway if it passed the permutation test. So pretty simple. Just look at the diagram and think of it. You will probably understand it better than I'm able to speak to it, right? So start with your pathways, find out what's in or out, then you actually measure an enrichment score, you record the maximum enrichment score, then make sure that that maximum enrichment score did not happen by chance, but you compare it with your permutated data set. So if it meets that criteria, then you can say, makes sense, I didn't get it by chance, so it truly has some significance, so let's focus on this pathway. There's nothing more to it. No more rocket science or brain science is involved in understanding GSEA. So that's the theory. And then some other words. So you can read that when I upload that. And what else I wanted to show you is I want to show you in our particular example how will we calculate the enrichment score. So I want to show you the formula, and this is also on your textbook. So the formula is we'll be actually calculating a z-test value for each of our pathway. And the formula for the z-test value is 1 over tk over k. So let me try to explain that in Excel. Maybe it will make sense. So what you will do is, so let's say this is your experiment that you did, and you had 10 particular probe sets or genes that had the highest expression. right? So these are the 10 genes that you have selected as the top expressed genes in your microarray. Now you are selecting a predefined sets of pathway 1 to 5, and you are picking them. As I said, you can pick them based on the KEGG pathway, or you can pick them based on the clustering you have done across experiments and earlier experiments. Then what you do, you construct a matrix, what we call is an incidence matrix. So what this incidence matrix will have is row wise it will have all the pathway, and column wise it will have all the genes that were differentially expressed based on a t-test or multi-test or SAM, FDR, whatever you may have done. So these are the 10 genes that were in my example, top expressed genes, column wise, and row wise you have the pathway. So now each pathway that are taken from KEGG doesn't mean all the genes that have top expressed will be contained in that pathway. But it happens to be, you mark it with a 1. In my case it has X. So by X I means that this gene 1 is present in pathway 1, gene 3 is present, but gene 2 isn't. So anywhere that I have an X here marks a presence of that gene for that pathways that I have selected. Then what you do to calculate the enrichment scores, I give you the example of pathway 3. So for each of this set, so each of the pathway belongs to your predefined pathway set. So you go through each of the pathway in your set and you calculate a reference T distribution for each of this pathway. So when you did your when you identified your top 10 genes, right? So each of these genes had a test statistics or a p-value to it. So in this case, I'm referring to the t-test, simple parametric t-test. So each of those genes had a t-test. So when you're calculating the t-distribution, the genes that are available on your pathway, you just basically sum up those t-tests and you divide it by the square root of number of genes that are available on your pathway. So in your scenario, you'll sum up 0.001, 0.003, 0.005, 0.003, 0.003, etc. And you divide it, the number of genes that are in your pathway are 1, 2, 3, 4, 5 by square root of 5. So that's your value of the Z. So t-test of each of the genes, and K is the number of your genes that are available on your pathway. Very simple. Then once you do that, since you started with a t-distribution and a bunch of statistics goes on to it, and end of the day, the Z values that you're getting for each of those pathway, right? So Z value for each of this pathway will also follow a normal distribution. So once that follows a normal distribution, then you will do a QQ plot to figure out, right, which are the ZK values that you have calculated are extreme. So if something is normal distribution and you do a QQ plot, pretty much every single value under the normal distribution will line up diagonally, pretty much the bar that you have here. But this particular data point here and this particular data point here, should I hold a criteria of -5 and 5 in my QQ plot, this is an outlier here and that's an outlier there. So these are the outlier which has extreme G distribution. So that's the pathway that actually stands out. And so each pathway has a Z value, Z distribution. Most of this pathway lies on this diagonal path. So end of the day, the pathway that has the most significance with the most extreme Z value is the pathway that's lining outside the defined criteria of -5 to 5. So you'll be picking up this pathway and that pathway. So as I work through the ALL data, so it will make sense to you what I'm doing. So that's pretty much all the theory that I have, and I will upload this. It may be already on your lecture 9. This slides takes you through working on the theory and talks about the central limit theory of why a t-distribution end of the day becomes a normal distribution. How do you derive ZK? But for me, what has helped me is just simply looking at this picture that I work with my choice of pathways. I find out how many genes belong to each of the pathway that I have selected. Then for each of the pathway, I calculate a score, whichever score based on the permutation stands up to be extreme, that pathway is the most significant. So I may have like 20, 30, 100 pathways. I have one experiment that I did. So which pathway has the most significance to my experiment? That's pretty much it. So this diagram helped me to understand initially. I read the paper that helped me, and then I also read this lecture from I don't remember where it was from. I think it was from Fred Hutchinson Cancer Research Center. And as you'll see, most of the authors of our book works in this cancer center. So they have enormous amount of wealth. Makes sense what they're talking and what they're presenting. So good materials to learn. So you have it on your lecture on Blackboard, so you can upload and review it again. So 524, so let's spend another 40 minutes or half an hour to actually go through the ALL code. So follow it how I'm doing it through the ALL data. So all that I will do, very simple. I'll just tell you to work with a different data set. I'll tell you to work probably with the same data set that you're doing with hypergeometrics. I'll request you to do normalization by VSN. So pay attention on homework 5 when you're doing normalization to VSN. You might run into some problem. So think through it, what you are actually doing, because some of the example that's given for VSN in your book is based on a 2-color array. Remember, you were doing an Affymetrix array, so think about it, what you are copying from book. And also the non-specific filtering, instead of IQR, I will probably ask you to do it from row SDS. Same thing. So basically you're going through all of your probe sets. Those probe sets that have a very small standard deviation means they're very close to each other. You'll get rid of it. Those probe sets that have a high standard deviation based on a cutoff, those are the one end of the day will give you low p-value, so you'll take those. Then I probably ask you to do a multi-test or a simple row t-test, and then I will tell you to do the gene set enrichment analysis based on you connecting to KEGG and picking up some pathways of interest. So those will be your collection. So let's do that with ALL data. Your homework will be the same thing except working with a different data set and different way of normalizing things. So you shouldn't find homework 6 for GSEA very challenging, like most of you are not finding homework 5 for hypergeometric challenging. But homework 7 will be a little hard because you have to understand linear modeling. So start reading Limma if you want to prepare it ahead of time. Okay. So enough on that. Just want to see if there has been any question posted. If not, I'll just simply go through the homework. To use telephone, because when we dial in, just hold. Let me see if I can, because when I went there, there was nobody on the telephone. So most of you are able to hear me from the computer, I suppose. Okay, let me dial in to the conference. So I'm in the conference now. Was there a question, Susan? No, I can't send IM from here. I don't know why DimDim has this music going on. It just really irritates me. Well, you can send me email if you have question about the project, because today I'm covering gene set enrichment analysis. So if you have a question on the project, just send me email. It's due tomorrow, and I'm going to hang up from the conference. It's due tomorrow, and I think one of the group J has submitted it. Group Y had a question. Only Group O, I haven't got any project proposal. So submit it by tomorrow. If you have any particular detail you want to know, ask me by email. All I said was also you can use minimum 3 data sets and run the MRA package or whatever package you want to use to do your meta-analysis. Nothing more I can think of right now. So I want you to be independent and do this work, because all the other homeworks on 7 I have given you as much guidance as I could, but this project will test that, after you actually complete this course and that's the first thing you're asked to do, how independent are you and how you're able to figure things out based on some of the homeworks that I have gone through. So that's the idea of the project. Okay, so let's get back to our gene set enrichment analysis with the ALL data. So I show everything with ALL because that's how the book has been done. So it makes sense for me to do that. So in your homework, you'll just work with a different data set. So we load the ALL data, and I will go through this very quickly because I have gone through that about 5 times before. Load the annotation. So that's my ALL data. So I bring the data as the data ALL. Okay, so let's look at they have different cell types, right? T cell and B cell. So I'm only interested on the B cells. So out of the 128 samples, I'm interested only on the B cells. And on the B cells, I'm interested on the BCR-ABL mutation and negative sample types. So you first have to get your cell types, and based on the cell types, you have to get the indication of BCR-ABL or negative. Okay, so I can actually get the B cell. So if ALL$BT gives me this matrix, so the way I can get the B cell is anything that starts with the B out of ALL-BT will give me the B cells. So I do a grep. Caret B means anything that starts with a B out of your ALL$BT, and this is ALL$BT. Now I am forming a collection of types to say further I'm only interested on those samples that are negative and BCR-ABL, and I stored that collection in types. Now out of your ALL data, if you look at the mol.biol, that's what actually shows you what are the sample types. So mol.biol shows you which is BCR-ABL or which is negative. Then you have other samples of P15, P16 mutation and ALL1, AF4 translocation, right? So I only want those sample types with BCR-ABL and negative, and I only wanted those samples with B cells. So the way I would then do that is whichever of the ALL$mol.biol has BCR or negative. So my collection was already given to be negative in BCR. So out of this ALL-mol.biol type, whichever is BCR equal to negative. So that's what I get that. And then from the main data set, I subset the one that intersects the B cell and intersects with your mol type. Okay. So I started with 128 samples, I think. So out of that, if I only take the BCR-ABL and negative mutation, then I should get 79. So let's just verify. Right. So now I end up with 79 samples. So these samples should be factored to say it only contains, as far as cell type is concerned, it only contains the B cell. So it doesn't contain the T cell anymore. So you have to establish a factor distinct on that. And it only contains the BCR-ABL and negative. So you also have to modify your mol.biol, have to modify your BT. So you do that. So this is just pheno data. In your example, it's much easier because you only upload a text file and you separate it by a semicolon or comma. So an ALL data is a little more complex how it has been constructed, but most of the homework that you have been doing is not as complex. So either your treat.txt, you separate things by semicolon and you read it out. So it's fairly simple, I believe there. Okay. So this is what end of the day I'll be proceeding my data set on, right? ACR-ABL with my 79 samples. So of those 79 samples, I want to do an IQR based filter, so to only get those that has an IQR variability of greater than 0.5. Anything that's less than 0.5 variability of IQR, I'll get rid of that. So I can do that by gene filter. And from homework 5 onwards, and also you saw in your textbook, instead of writing the gene filter and instead of writing a function of IQR, you can actually use a predefined NS filter function, which you can do a variable cutoff of 0.5 easily. It has additional parameters, like you were doing in homework 6.5, of retaining only those probe sets that have entrance ID, removing those that has a duplicate entrance ID, only containing those that has a biological process of any of those go process or molecular function or biological process. But I don't ask you to do this, so I wouldn't do that too. So only do that. That's for the IQR of greater than 0.5. Other parameters, I'll leave it alone. So now, if you look into the what does NS filter returns, so the way that function has been constructed, the NS filter returns the subset of the expression object, right, which is $ESET, and it tells you how many samples and others. So you don't have to do an array and subset it from ALL. It's already returning you a subset of ESET. It tells you the different things from the filter.log, what it has removed or not. But in our case, we actually haven't done anything for removing entrance ID or biological process. So only criteria was remove everything that's less than 0.5 or retain those that are greater than 0.5. So what it has done out of those features that we started with, 7,000 something, in the log file it will tell you 4,400 that it has removed, which did not meet the IQR criteria. Okay, and then it also removed the 889 for the entrance ID and the duplicates, but it didn't do anything with the biological process for the go, because we didn't give it in the parameter. So that's your filter negative. So then you started with your ALLBC, this was the one that you started with, that return. So if you just $it with your expression set, so that's already going to be your reduced feature of, 4,399, right? So you can just say filled at BCR negative $ESET. So if you just reference it with a dollar, you can construct your subset of the expression set that will only contain this 4,399. Little different than how I've showed you in the past. So my BCR ABL negative after the IQR filter criteria, right, ends up with 4,390 feature and removing the duplicate for the entrance. But I did not do anything with the go here. So 4,399 feature is the subset of what we started with. Now the fun begins. So now what I want, now this is the main part of the go, right? Pre-processing ahead of this. So now what I want to do is I am importing everything that, so making a connection to the KEGG library. So if you don't have this library, you have to do a BioC to get it. So that enables me to get all the pathways that I want from my bioconductor. So I already had that before, so I'm going to load this annotation or that has a source of KEGG pathway. That's why I prefer working with bioconductor. Just from one portal I can connect to many different data sets, bring everything that I want into my single workflow. Otherwise you have to go and manually go through all those different websites and download and write scripts in Perl and whatnot. But anyway, that's another way of doing things. The main library that you want to work with for your assignment and this example also is the library GSEA base. Again, if you don't have it, you have to BioC and get it, right? So here you are actually constructing a collection. So what this particular function will do, gene set collection, it will collect all the pathways that are available for KEGG. So get all the pathways, and at the same time it will also remember what are the different genes that were selected on your ALL fit BCR negative. So it will remember the 4,399 features that is relevant to your experiment. In addition, it will import every single KEGG collection that it can get from the database. So within this subset of parameter, if you want to subset and say further reduce the list, it may have, I don't know, 500, 600 different or maybe more pathways. But if you're only interested to work with 10, 100, however number of pathways, you can actually give additional argument here. But for this example, I will bring everything that's available in KGG. See how many it brings. So if you look at it, it will bring in, based on your data sets, it will bring in 210 pathways, and these pathways contain 178 unique features. So that's exactly what it did. So these are the different pathways. The number of them is 210 pathways that it brought in. And if you were to get all these distinct features that were available, so you may have on your experiment 1700, 1800, whatever you have, right? So that's on your ALL BCR negative. So in your experiment that you did, you have about 4,399 features based on your IQR. So you have 4,399 of them, but your gene set collection has brought 210 pathways where at least one of those genes are available. That's available on your list of 7,000 is also available in this pathway, and it ended up importing about 210 pathways, and each of them has 7. And when you put all this excess to see at least one gene is available in that pathway. So this column is from your experiment, and these are the pathways you got from your KGG database. And if you were to get the distinct of all those excess where there's a match between gene on your experiment, which is 4,000 something for ALL subset, and 210 pathway that you imported, so all the distinct features that belongs to all these pathways in this collection is 1758. So your ALL BCR negative may be 4,399, but the 210 pathways that you just imported from KGG, if you count for all the excess where there's a match, there's 1758 of unique features. So now comes the big part of constructing the incidence matrix. So as I say, the incidence matrix is columns will have all the probe sets that's on the subset of your ALL BCR negative data, which is all the 4,399 features, and the rows will be the pathways, 210 of them, where you just imported from KGG. Whenever there is a match, that's an X. You're not going to import a pathway from KGG where you didn't have a single match. That's why you had 210. But let's put it out on the incidence matrix. So you got your GSC, so you can wrap it with the incidence. So this is already pre-built for you. You can copy the exact same code. So once you wrap it with the incidence, it will construct the incidence matrix for you, and you can assign that matrix into your AM. So you cannot visualize properly here. So these are the different pathways that you have, and on the column wise you will have all the genes. Whenever there was not an intersection, there was 0, meaning nothing. Whenever there was a match, which was X in my example, there would be a 1 here. And you can actually calculate all the ones, then you can figure out how many genes actually mapped. So in my example, if you had a 1 here, instead of X, I write a 1 here, and so I will have 1 and 1 is 2. So if I do a sum row-wise for pathway 2, I can tell I had a match of 2 genes that was in my experiment with the pathways that I just imported. So pretty soon you'll see the significance of a row sum. And anywhere that the gene is not present, the incidence matrix will have a value of 0. So when you visualize it first, you see all these zeros because there hasn't been a match. Whenever there is a match, you'll see a 1. So I hope you got the idea of incidence matrix, right? So you can very quickly see if you go column wise, right? And if you this is a matrix, so if you do a column names on the matrix, it should give you all the probe sets, right? So column wise you have probe sets, right? So you got 1758 probes that are matching that's in your pathway, that's also on your gene sets, on your experiment. So that's 758 of them. So these are the different probe sets that's in the pathway, and that's also on your experiment. And if you go row wise, it will tell you all the pathways that you brought in. So if you do a row names, right, then you get all the pathways. So now it's a matter of figuring out each of these row, what genes are in common, and then get of all those t-tests that's available for those genes and calculate an enrichment score. And whichever pathway comes up with the highest enrichment score in compared to your permutation, that will be the pathway that stands out. So that's where I'm going end of the day with that. So hope this makes sense, because that's once you get this concept of how you got your pathway, how you got your genes, where are the ones, where are the zeros, how you're calculating your score, and then you'll figure out how to do gene set. Okay. So I got my incidence matrix. I can go column wise to figure out all the probes. I can go row wise to figure out all my pathways. And now what I should do is this ALL-BCR negative, right, that I did had 4,399 features, but on my pathway I only had 1,000. How many did I have? 1,758 features. So no point on working with the features. So no point. So if you had another feature here, so let's say gene 11, I don't know, and none of this pathway had a X in it, that means this gene is not present in any of this pathway, no point bothering about this gene anymore, because it's not going to make any difference to any of these pathways. So what I do is then from my main ALL-BCR negative, I only get those probe sets. So column name AM already gives me those probe sets. So I only get those probe sets that are available in my incidence matrix that had a cross with my pathway. So I subset it. So now if you look at your NSF, it will match 1758. That it does. So now your reduced set now is 1758, which had a cross with your pathway for those 79 samples. And then this NSF is the one that I will use to calculate the test statistics, and they will come into play to calculate your Z-score. Now some of the questions I may ask in your homework, like how many gene sets and how many genes are in your incidence matrix. So if you do a dimension, so then you can say there are 1758 gene sets for my 79 samples. Okay. Then if I ask you how many gene sets mean how many pathways, right? So pathways you can easily get to by column name row names, 210. So I'll ask you in your homework on your incidence matrix, how many pathways did you get? How many distinct probe sets did you get that matches with your pathways? Then let's say how many genes have fewer than 10, how many gene sets have fewer than 10 genes, right? So what I mean by that is if you look at this pathway, it had a 1 here, it had a 1 there, and rest are all zeros. There are only 2 genes that matched here. In this case, it will be 1 here, 1 here, 1 there, 1, so 4. So if you look at your particular example, you have about 1,758 this way, and you have all these pathways. So I want to know which rows where you minimum have 10, right? So if you end up summing your rows, then you will get the value. So if the sum is greater than 10, then you know for sure on this row you had at least 4 genes that match with your pathway. And how would you do that? Right? So first of all, if you do a row sums, right? Very simple. You have a matrix. So if you do a row sum, it tells you on each of this row what's the sum. So if you do a row sum for pathway 1, your row sum is 4. For pathway 2, your row sum is 2. For pathway 3, your row sum is 1, 2. X means it's there. So 1, 2, 3, 4, 5. Right? So that's exactly what these row sums are doing here. So for each of this pathway, it tells you there's 47 genes present. This pathway 12 genes present. This pathway 7 genes. So if I'm asking you what are the pathways that have less than 10 genes, so if you do your row sums, just by looking at it, you can say these will be the gone because this has less than 10, this has less than 10, right? So you can just basically look at your entire matrix, which is your AM incidence matrix, and you can subset it in your array for all of those that's less than 10. So if you store this into n gene, where all the row sums, so if all the row sums, right, just like I showed you, are stored in your n gene, so anywhere the n gene value is less than 10, then you can again subset it out of your row names AM. There's a lot of subsetting goes out of array from R. I can't explain this in words, but try it out and try to make a mental map. What was the main array matrix and how are you subsetting things? Okay, so these are the particular probe sets. So for example, I had an example here, 4614. You'll probably see 4614 here somewhere. That was less than 10. So these criteria is met. So those are the different pathway IDs that had at least less than 10 genes mapped to it. You can very easily get to it. So that will be probably one of the questions I will ask you in the homework. I may ask you different. I may ask you give me everything that's greater than 20. I don't know, something like that. So then you know what you can do. So since these are the genes that belong to each of these pathway, you can actually sort this matrix. So which will be very interesting. That will tell you the pathway which has the highest number of genes mapped to it. And if you sort it decreasing, so 67 will be there, followed by, I don't know, followed by 39, 25, 12, or maybe 100 will be the first one. So you should be able to sort it and say which pathway ID has the highest number of genes mapped to it. And you'll sort it. So n gene has all the connection between pathway to number of genes mapped to it. So if you sort it by decreasing equals to true, you'll get everything, right? So of this everything, the first value, since you're sorting by decreasing, will be the one that has the highest number of genes. So 100 will be by 90, then will be 80, then all the way to 1. So you can easily point to that by indexing the very first one. So this pathway ID 001100 has the highest number, 407 genes that are mapped to this pathway ID. If you get to the second one, you can very easily get, you can very easily get the second one too, just to index the second to see what's the second one with the second highest genes mapped to it. So that will be your pathway ID 5200 that's mapped, that has the 165 genes mapped to it. Then it's beautiful and you'll like it that, then let's look at what's the name of this pathway ID. So this is already figured out into your annotation. So you go from KGG pathway ID to the name of the pathway. So you supply the, when I showed you this one, the pathway with the highest number of genes has an ID of 1100. So you supply that in this parameter and they'll give you the name of that pathway. So metabolic pathway is the pathway with the highest number, about 407 genes that you had across in your incidence matrix when you lined out all the probe sets of your subset is ABRBCL data against the pathways that you have. Okay, I think we're good so far. We'll spend another 10-20 minutes to wrap it up. There's no questions. It should be very simple, these things, once you practice it. Now what you want to do is you want to calculate the test statistics, row t-test. So row t-test is you can question row t-test and see how it's calculated, and it's also on your book. So you need to know your class label, right, to do this t-test, and you supply the data set that you want to calculate the t-test. So now all you are doing for each of the genes that you have on your subset of data, you're calculating a t-test for all of them. So your mol.biol gives you all the class label, right? So you have all the BCR, ABL, and negative. So you already factored it before, so you should work correctly. So these are the different class labels. So on your row t-test, you call this function, and NSF and NSF was a 1758 features. Remember, you started with your BCR ABL of 4000 something, then you constructed an incidence matrix, and you did a subset of 4000 something based on your incidence matrix, wherever those probe sets where you had at least a match with your pathway. And when you imported your 210 pathway, you also imported 1758 unique features. So that was the last subset that you had, and that's now with your 1758 features. You want to calculate the row t-test and you give them the label. So now if you look at this, it will output two things, the row t-test. It will output each of these test statistics, and it will also output the p-value. So we probably want to, so what we said in our example, if you look at the formula, we want to work with the t-statistics. We're not interested in the p-value. Right? So we want to sum each of the test statistics that's given for each gene that belongs to your pathway, and we want to divide it by square root of the number of genes that's in that pathway. That will give me my z-test, and that z-test will be the criteria that I'll finally use to say which pathway I want to keep and which pathway doesn't show any extreme value, so it's not related to my experiment. Okay, so I also want to remove from my incidence matrix, if I did not have more than 10 genes that mapped to my pathway, I can also give that criteria. So as you saw, the highest number had about 400 some genes mapped to it. The second one had 100 some genes mapped to it. So if you happen to have a pathway that had very small amount of genes mapped to it, you can take all the t-test of these genes, your z-value will be very small. So it wouldn't make any difference. So you rather focus on those pathways that has a higher number of genes mapped to it. So when you calculate this enrichment score or running sum, obviously the pathway that with 400 genes will have a higher running sum than a pathway that had only 10 genes or 5 genes. So you don't need to bother giving those pathways. So I might give you a different criteria, but you can very easily get rid of those pathway that has a greater than 10. I might tell you to give greater than 15 or greater than 5, something like that. So that's how you do. So you would again subset it, right? So now that you have that, let's calculate the zk. So again, remember the zk formula, sum of all the t-test over square root of k. So what you need to do, your RTT that you got, right, when I showed you, RTT contains your test statistics, your RTT. What are the different things? RTT contains test statistics and p-value, but I'm interested in test statistics. So if I dollar the RTT with statistics, then I will get all the t-value. So let's fetch only the t-values. You cannot just use RTT. You have to use t-test. You cannot use p-value. So be mindful of that. So now this RTT stats that I have contains all the test statistics for 1756 genes. So these test statistics will be feeding in my formula here of TK, and K I already know. Wherever there were ones, right, that means there is a match between gene to pathway, that's my K. And that's very simply how you calculate the ZK for each of these pathway. Okay. So then the way I do that is whenever I have a match of my t-value that's also available on my AM2 matrix, and AM2 is where I have at least more than 10 genes that had a match with my pathway. So I calculate the TAs. So that's my TA. And then I divide the TA by square root of row sums AM2. So if I do a row sums AM2, that's the K, right? So if I do a row sum, so 1, 2, 3, 4, so that means 4 genes mapped to a pathway, so that's K. So square root of K I already got by my row sums. Square root of K I got my row sums, and TK I already got from here, from my test statistics, where it was actually matching with my AM2. And AM2 are only those subsets that had greater than 10 genes matching to the pathway. So that's the formula for T adjusted, so which is also your ZK value. So I calculated the ZK or T adjusted, and I can get the names of the IDs of those pathways. So for each of these pathways, now I have a z-test value. Okay. Now, as you know, the z-test value that you got for each of those pathways is also normally distributed. And why it is normally distributed and etc., it talks you the central limit theorem. So take their word for it that all the z values that you have, since you started with a t distribution, the z distribution that you will have will also have a normal distribution. So if it's a normal distribution, then we can actually draw a QQ plot. So t adjusted is all nothing but your z value. So let's draw a QQ plot of this, right? And let's just figure out a line which it will map this QQ plot diagonally. So you can do a QQ line. Where's my script? Okay. So now if you do a QQ line, so as I was saying, you got your z-test value for each of this pathway. That z-test is normally distributed. So you draw a diagonal line. So for a normal distribution, you will see a pattern where most of the z-tests for each of this pathway are on this line. So I'm only interested on those ones that are outliers. That means those z-tests are extreme. So those are the only pathway that I'll pick. So if you look at this particular plot dot here, so that seems like an outlier, and these are also seeming like outlier. These I'm not interested. They are not extreme. Okay, so if this is -5, so this is probably -6, and that's probably -7. So out of this Q plot, I can get the z-test that's maybe greater than -7, and out of this T plot, I can also, this is 5. So if I line it up here, this is 5. So I can also get the z values that are greater than 5. So what it will do, it will fetch me from my QQ plot these 6 of those, I think 1, 2, 3, 4, 5, 6. That's greater than 5. And for -7, it will probably get me, or you can also do -6, it will give you 2. If I give you in the homework a criteria of -5, it will give you 3. So these criteria I will change. I will tell you from your QQ plot, do something greater than -5, or I can say do something greater than -7. So you have to understand the scale and what I'm asking, what you're supposed to filter. So let's do greater than -7. It should get this one. So if you have a t adjusted, which I already did it in the Q norm, of less than -7 of the QQ plot, it should give me the probe set ID of SMPW. So let's unlist that and see what pathway that SMPW belongs to. So SMPW is pathway ID 3010, and the value it also gives you is -8.76. So if you go back to your QQ plot, so this one has a value of -8.76, so which met my criteria when I wanted to get it out of greater than -7. That was the pathway ID 3010. So let's now use the annotation to get the actual pathway name. So what's actually showing you here, ribosome pathway, is based on your QQ plot on the lower end is very extreme. Then these are also different pathways that could be extreme. So let's write greater than 5. Same thing. This one I did less than -7. So let's get this greater than 5. I should have more of those. So if I do that, I got this many. 1, 2, 3, 4, 5, 6, 7, 8. So probably gave me 1, 2, 3, 4, 5, 6, 7, 8. So this is probably just a little over 5. Then again, you can select this to see what were the exact cutoff values and what were the pathway IDs by doing your log PW here. So this will give you the different IDs. So just barely 5.1, so it met the criteria. So maybe in homework I'll give you a different criteria, how you want to cut your QQ plot, and then you get your pathway IDs. Then I can tell you, just pick a pathway ID and do a heat map plot for that pathway ID. So here is an example. So I can just get a particular pathway ID, and this was my data set that I started to, and that's my annotation. So what's that's actually showing you, if I'm like focusing on a particular pathway ID, so these are the different probe sets that belongs to that pathway ID. These are the samples, so sample versus probe sets, and this is the expression pattern. You can look to see for these genes has a little darker red color, has a lighter red, yellow color. So are these down regulated and this is up regulated for this particular probe sets for this pathway ID? Then those of you who are biologists can answer a better question. Does this make sense? This pathway ID for this kind of gene expression that you're getting. So I feel it's pretty neat. I think this makes much better sense. Then the final topic of gene set enrichment is just because you got a pathway a G-score of minus 8.7 and it was an outlier on the QQ plot does not mean that you did not get it by chance. So like we always do in statistics, that we permutate the class label and we try to see if I were to randomly permutate and would I have random based on those probe sets that I have selected in my microarray, would I have just by chance got a pathway ID with such a small value of minus 8.7. So that we just then compare it with a permutated data set, and in order to do that you have to import the function category. This is a random seed 1, 2, 3, depends on, it's a seed you give to randomize your value. And then you calculate the p-values by using this gset perm. So this actually will be permutating all of your class labels, and this is the class label that you already know, right? So if your 78 samples, this is BCR-ABL, this is negative, so what it will do, it will make this one negative and this one BCR-ABL, etc., etc. It will do a permutation of 1,000 times. AM2 is the matrix where you have all of your pathway to your gene sets. Then for each of those particular pathway and each of this row, it will actually calculate a p-value to say, does this, so now, okay, let me show you the p-value for each of those pathways. So let's look at the top ones. Sorry, it hasn't finished calculating. So this pathway has a low p-value of 0.14 and a high p-value of 0.851. So for each of these pathways that you have, by permutating the class labels, each of this pathway will get a p-value calculated. It will have a low p-value and a high p-value. And I give you the definition of low p-value, where the proportion where based on your permutated sample, you have p-values that were actually smaller than what you got observed. And the upper one is where you got p-value that's higher than you got observed. And that's where you actually will establish the criteria. Now that for each of your pathways you got a p-value, you're interested on the lower end where you got actually lower test statistics that's less than what you observed. So you can get a cutoff value of less than 0.05, and these are the pathways that met this criteria. Okay, and then you only want to use the low C, which is you had a low one and a high one, and let's unlist those pathways that met your low C criteria. So these are the pathways: glycophospholipid and RNA polymerase, mismatch, etc., etc. So that's pretty much end-to-end where you will start with your data sets that I give you. You probably want to do a VSN and a row SDS, just like the way you did in hypergeometric. Then I will ask you to connect to KEGG database. Then you will run your gene set enrichment collection. You'll calculate your test statistics, your Z statistics, and then you will do a QQ plot. I'll give you a criteria of what's actually on the low end and what's on the high end. Then you will actually pick those particular test pathway ID and compare it with a permutation to say, did that also meet your criteria or not. So I will upload this exercise that we just did. Then you also need to study chapter 13 of your textbook and the paper that I told you. So I will upload the homework hopefully by tomorrow on Tuesday, and it will be due in 2 weeks. So that will be a gene GSEA. Your hypergeometric test that you're working on now will be reviewed next week. And we also, as a future topic, is Limma, which is linear modeling for multiple time points, right? So when you think of it, right now when you do your analysis, it's a single experiment done one time. But what if you had an experiment where you took samples at 1 hour, you took samples at 10 hours, then you took samples end of the day. So you might be interested to ask the question to say, okay, what's the difference when I did this experiment 1 hour versus when I did it at 24 hours? What genes are always upregulated between 1 hour to 24 hour? I might ask you a question, okay, what genes that were upregulated from 1 hour to 10 hour, but were downregulated from 10 hour to 24 hour. So you have to think and analyzing and modeling your data based on the different time points that's within your experiment, because to this date you're only focusing on one experiment. So your Limma will expose you within an experiment multiple time points, and the project that you're working on is a meta-analysis, which is you have multiple experiments, and then you try to see the different genes that are individually showing up across your individual experiments that's top expressed. But if you do a meta-analysis, how does it relate for across experiments? So those are the final two topics of linear modeling, which would be homework 7. Meta-analysis, you're already working on your project. Hypergeometric test, homework 6, homework 5, right, will be reviewed tomorrow. So I wanted to expose you the best I can to gene set enrichment analysis. This particular exercise that I did will be uploaded tomorrow or today. And I will also try to upload the recorded session and expect to see the homework 6 loaded next week. And if you have additional questions on projects, send me an email. If you have additional questions on the grade that you received or whatnot, try to send me a question. And one thing I wanted to clarify about resubmitting of your homework, I don't recall I ever say that I'm just going to keep inventing new challenge and homework for you and you will be resubmitting. As alone, it's hard for me just to construct one set of homework. But I think I may have indicated that if you're really not happy with your homework, you can work with a completely different data set. That should be the first point. And then you have to do what was asked to do. Then you have to additionally tell me what is that you did different, right? That makes me believe that you have a good handle on the topic. So those initiatives has to be from you. You have to write a mini analysis or a mini report for me to convince me that it's worth for me to go back and change your grade. If you just took my solution and rewrote the same and reposted the same, that's not worth my time. That's not worth your time to engage in an upgrading or downgrading your grade. But I'm not having that issue with most of you or any of you. So I'm not saying that that's the issue. But if there was any misconfusion, I wanted to clarify. So good job so far. I have seen so many of you progress so well and so good that makes me feel good. And those of you who have still some additional question, reach out to me. We only have a few weeks left. So let me stop the recording, and I don't have anything else to cover. So have a good weekend, and then we'll touch base next Sunday. Hopefully, maybe next time, if it works, most of you, 5 o'clock, so we can enjoy the day during the daylight. And if not, I may want to schedule it earlier. So that's it. Have a good night.